Hey, Mr. YS here, another tutorial on D2 digestion. Alright, so have a look at the pizza. If we look at food, think about food, then we're having some impact on our digestive secretions. So we're going to have a look at how are digestive secretions controlled in the stomach. Why is that important? Why would it be important that we don't secrete things all the time and we can control when and if we secrete them? Well, if we were to secrete all the time, it would just be a waste of enzymes. So we don't want to waste. It would be the issue of wasting and the energy to put into it. So we want to control when we secrete and how we secrete. And we do that by really secreting when we see food or if we taste food. Those are going to be times where, now we're going to secrete things in the stomach and, in fact, throughout the rest of the digestive system as well. Okay, so we mentioned a couple of things then. So then how does the body actually do the control? Talked about seeing, tasting. What mechanisms could it use to control those secretions? Just stop and have a think. Well, if we're controlling it through seeing, tasting, and maybe even thinking about food, even dreaming about it, then we're involving the brain. And if we're involving the brain, that is to do with the nervous system. So we've got the nervous system is starting things off. And then the nervous system has got to have a way to send signals. But the other thing that can be involved, and often is involved, is the hormonal system, which is about going through the blood. So the two ways are the nervous system and the hormonal system. So we get nervous and hormonal mechanisms controlling the secretion of digestive juices. And there might be times that we want to switch that off as well. So if we are in a fight or flight situation, then we would reduce activity in the digestive system. If you've ever been really nervous or really stressed, people can go for a long time without eating, without feeling hungry. And then at other times when we want to do it during rest, we then increase the activity of the digestive system. Okay, so throughout the digestive system, we have exocrine glands. They are, and we're going to have a look at what they are in terms of their general structure before we get into the stomach in particular. So an exocrine is a exocrine gland, but exo means outside of, and crine means blood. So it's outside of the blood. These are glands that are going to give secretions down into ducts, but not into the blood. And so the digestive system, the whole thing is the alimentary canal. But the alimentary canal is not in the blood, it's outside of blood. So any secretions into that are going to go there. So stop and think if you can think of 3 examples of different exocrine glands and what they secrete throughout the digestive system. Okay, we'll come back to that in just a minute, but let's have a look at the structure of one of these glands then. So we see that we have cells arranged in a kind of almost a complete circle around a central canal. And so let's try and reveal these. So we've also got this blue layer. I mean, it's not blue in reality, but the blue layer, which is kind of surrounding, it's like a coating around them. And so this is the basement membrane, and this basement membrane is going to stop things leaking out. So that means everything's going to be channeled down through here and out that way. Okay, so basement membrane, its function is to kind of enclose these cells, and we can see lots of cells arranged around. And so these cells are our secretory cells, and whatever that would be secreting. And if we have a look at what's inside them, well, if it's doing a job of secreting things, it means it's making a lot of proteins. So just stop and think about what these things could be that it needs a lot of if it's going to be making a lot of proteins. So what it will need a lot of, it will need a lot of mitochondria, because it needs ATP for energy for making all these proteins. It will need a lot of vesicles to transport proteins out via endocytosis. And it's going to need a lot of endoplasmic reticulum, the rough endoplasmic reticulum, because that's for transport outside of the cells. Okay, down to over here, and we have a duct cell. So this central bit here is the duct, and the duct is going to pull the secretions to wherever they need to go. So it could be down into the small intestine. It could be into the mouth. Yeah, so we'll have a look at some examples of those. All right, so these are surrounding these cells, the whole thing we call an acinus. So it's an acinus of secretory cells surrounding the duct and, well, yeah, creating the whole gland itself. So there's our question there. What is an acinus? It's a group of secretory cells which make up exocrine glands. Okay, let's go down to a real picture here, and I want you to see what you can identify. So just stop and see if you can identify some structures that we saw up there. Okay, a little bit difficult to see here, but we can see certainly the duct leading down here. So it's kind of pooling that way, and then we don't see exactly where it goes off. And then we've got the ring there. That's the whole thing being the acinus. And we could label the outside of that as being the basement membrane. And each one of those is a secretory cell. So we've got secretory cells, the duct, the basement membrane, the whole thing being an acinus. And yeah, those are the structures that we can see. The rest of these are just cells of wherever this particular acinus is. Again, see if you can now label some of those parts. So stop and see if you can apply those labels. And what we have here, we've got our secretory cells. We have a whole acinus. We have the duct. And we could call this a lumen because it's the hole in the center. And then down there, surrounding the whole group of them, that's our basement membrane. Alright, so zooming into one of these individual secretory exocrine gland cells, just stop there and see if you can identify the different structures within the cell. Well, over here we have a nucleus. Down here we have lots of mitochondria. We have over here rough endoplasmic reticulum. And although you can't really see it pointing to it there, I think it's probably talking about ribosomes. Now these look very similar. So over here, we can't see wiggly lines in there. We don't see this kind of, you know, the wiggles of the membrane. So I think these are then the vesicles, the secretory vesicles. Okay, here we are zooming in to a slide of the stomach. And so we can see there are different layers, a bit like in the small intestine. So let's go through and have a look at what these different parts are, because we're going to think about secretions into three areas, and in particular into the stomach. So in the stomach, we've got these dips. We can see that it goes down. There are these dips down, and they're lined by cells. Now these are called gastric pits, and we're going to have a number of different cell types making up these gastric pits, and we'll come back to those cell types. But they're then secreting things into the stomach. So these are going to secrete digestive enzymes. And we're also going to see hydrochloric acid, HCl. Okay, so what else have we got? We also have, as part of those gastric pits down at the bottom, we've got gastric glands and the cells which are helping to produce the exocrine substances. We then have a layer of muscle. So the stomach has a muscle layer to help it to contract and to churn the food. And this whole thing, just like in the small intestine, the bit where we actually have the functional bits, the last layer is the mucosa. And then underneath that, you've got submucosa with blood vessels. Okay, back to that first question about naming three places where we have exocrine glands that secrete things into ducts. Well, the first one that we've thought about already is salivary glands. We've got salivary glands, and they release water, salivary amylase, mucus, and they also release this enzyme, lysozyme. This is also released in the eyes. It's a defense against bacteria, so it helps to break down bacteria, break down pathogens. And the other thing that's in it is electrolytes, which we can think of really as being kind of salts. Okay, so there's water, salivary amylase, there's mucus. This is for helping to lubricate the food as it goes down. Okay, the next place that we had something that we'd already talked about was pancreatic juice from the pancreas, which had all the enzymes. So that one's quite an easy one. We can think about all the enzymes, and we give them the very specific bit here. So it's got pancreatic amylase, pancreatic lipase, and we talked about trypsin. This is what it is when it's activated. So trypsin, which was our protease. Now also, because it's neutralizing the acid, we've got bicarbonate ions. And this is to neutralize acid as it goes into the small intestine. Okay, and the final one, which we have thought about as well, and we're going to do in more detail today, is gastric juice, which is same things. We've got water, mucus, we've got enzymes, and so the main one is pepsin for protein digestion, and we've also got renin, which is also involved in protein digestion. All right, so what happens then when we see food, we smell food, and we are preparing to digest? Well, we get a signal, and the signal of food, we have the input to the brain, to the back of the brain, to the visual cortex, and then it sends signals to the region of the brain that is it's subconscious, it's autonomic, we can't control it. And here it's the medulla of the brain, the medulla oblongata. And it's at the base of the brain in this region here, near to the hypothalamus. And the medulla is then going to send signals. So the other thing which we could have happening down in the stomach, and I've just drawn it as a circle there, is we can get stretching. So if the stomach stretches, if you eat, then that can send signals back up to the brain, and then the brain sends signals down to the stomach. So over here we've got the stomach is going to be involved, and the stomach then, under stimulation from the brain, it will release a hormone that we're going to have a look at, gastrin, and that's going to cause the production of stomach acid. And so the brain can also directly signal to cause the production of stomach acid as well. And then we get this hormone gastrin that we'll have a look at in a moment. Okay, we also have the small intestine. And the small intestine, it can be involved. It can release some hormones. And these are like a level 7, high level 7. It releases secretin and CCK. And those are involved in causing more pancreatic juice and bile to be secreted, but also it goes back and it kind of switches off. So this is the thing that we'll look at. It switches off stomach secretions. So we'll come back and have a look at that. Right, let's have a look at this in a little bit more detail then. So we had the idea of there's a stimulus, and the stimulus could be the smell, could be receptors either in the eye, it could be on the tongue as well. We've got receptors in the nose, on the tongue, in the nose, in the eye, and all these receptors are going to send signals, and then either through the visual cortex or the other sensory cortexes, we're going to get signals sent to the medulla. The medulla is then going to send a signal down through nerves. And the nerve that's involved here is called the vagus. Not like Las Vegas, vagus nerve. And so this then is going to connect with cells in the stomach. And we'll have a look at that in just a second, where it's going to connect with cells, and then we're going to get the release of hydrochloric acid. We're going to get the release of pepsinogen. And the other thing that will be released as well, and it's going to go into the blood vessels which are surrounding and over. So there are loads of blood vessels covering the surface. We're going to get the release of a hormone which is gastrin, and it's going to go into the blood, and it's going to travel around, and it's going to affect the cells in the stomach. So these signals are going to affect the gastric gland that we can see here. Here's a gastric gland, and the gastric gland has different cell types in it, and they're going to release different things. So we've got some surface mucus cells. They're going to release mucus. This helps it's going to protect your stomach, protect it from acid digestion, and also from protease digestion. Further down we can see that we have Mucous neck cells, not really that important for us here. We've got parietal cells. And so knowing these is quite a high level thing, but it definitely comes up in all the mark schemes. So I remember parietal, P for pH. And what pH does the stomach have? It has a low pH. And what causes the low pH? It's H+, and then, you know, it goes with chloride ions as well. It's the hydrochloric acid. So parietal cells cause low pH by secreting. They're going to put the HCl into the stomach. Here we have got an endocrine cell. Now, if it's an endocrine, it's actually secreting into, not into that bit there, it's going to secrete into blood vessels. And so if it's secreting into blood vessels, then the thing that it's going to be is it's going to be secreting gastrin. These are called G cells. So G for gastrin, G cells. G cells secrete gastrin into the blood. And down at the bottom there, we can see we've got our chief cells. And so the chief cells, it's the chief enzyme, the main enzyme that we think about in the stomach. They secrete pepsinogen, which is then, by hydrochloric acid, well, because of the hydrochloric acid and the low pH, it's converted into pepsin. And now we have our active enzyme. But what's actually secreted is the pepsinogen, and then it becomes the pepsin. So we talked about all the things there. We get mucus secreted, we get hydrochloric acid, we get pepsinogen, and we get this hormone gastrin, which is going to travel via the blood. So it's going to travel via the blood system, but it's actually going to then, because the blood system is going close to all of these, it's going to affect these other cells. And in particular, it's going to affect the parietal cells, and it's going to affect the chief cells. So it's going to get over to those, and it's going to cause them to increase what they're doing, what they're secreting. We'll get increased HCl and increased pepsinogen. So gastrin is kind of like positive feedback. In a way, because it's allowing more release of these digestive secretions, and it's being caused by seeing food. So it's kind of a positive feedback to get more secreted. And also stretch receptors would lead to more of its release. So that's a kind of positive feedback. So in here, although we haven't shown them, there will be receptors, and they can detect whether the stomach is stretching. And when it stretches, we get more gastric juice. So what is gastric juice? Gastric juice is all of these things. It is the hydrochloric acid, it's the pepsinogen, it's the mucus. All of those things are gastric juice. Okay, here's our quick recap summary. So if we were going to write something on this, it could be up to 6 marks about how gastric secretions are controlled. Well, they're controlled by both nerves and hormones. So we talked about the vagus nerve from the medulla of the brain. If we have a stimulus like the sight or smell of food, it's going to cause us to secrete gastric juice. And that was by sending signals down the vagus nerve to the stomach, causing the release. We also have in the stomach, we talked about stretch receptors, and the other type is chemoreceptors. What they can detect, they will be detecting things like peptides. So in a way that they're detecting protein. So they can detect what level of protein is in the stomach, and that might lead to more secretion of digestive juices. And then stretch receptors, as the stomach stretches, it causes signals to go back up. So it's a two-way communication. The brain is sending signals down to cause release of gastric juice, but also the stomach can send signals back up to the brain, and then that leads to more signals coming down the vagus nerve. And then we get secretion of gastric juice, and we get secretion of gastrin, that hormone, and that's our hormonal control, by the G cells. And then the gastrin leads to more secretion of gastric juice, more secretion of pepsinogen. We said it was by the chief cells, hydrochloric acid, low pH by the parietal cells. And then we did mention before briefly that when food goes down into the small intestine, so once it's left the J-shaped pouch, once it's left the stomach and it starts to go into the small intestine, its presence there is detected by receptors, and then that will lead to the release of these two, secretin and somatostatin. And they can go back, and so this is a very high level statement, they can go back and then they can inhibit. They will inhibit the secretion of gastric juices and kind of switch off the action of gastrin. Okay, so once we've got these things secreted, one of them is stomach acid. So what's its role? And we've talked about it before. Stop and think about what is its role. Can you remember where it was secreted from? And what its consequences are? Okay, so quick recap again, because it's a bit of a repeat. It's low pH, it's the parietal cells. And what it does is it denatures proteins. So we've looked at the idea that protein structure is complicated, 3D. It's got these helices, we've got these beta pleated sheets. And when we get low pH, those protons are going to go and interrupt the bonding, and we're going to get denaturing. And that, when it's denatured, it kind of loses its shape. It's just going to lose its shape, and that helps because it exposes the polypeptide chains, and that means that our enzyme, which is now active, pepsin, can start to make cuts. It will start to cut it'll make cuts in those chains, and now we're going to get those shorter polypeptides. And we said that pepsin was released by the chief cells, but initially it's inactive. It's as pepsinogen. And so the acid environment helps to convert the inactive pepsinogen into the active pepsin. So how do we know any of this? And here's where the IB's put in some kind of historical stuff. How do we know about the chemical nature of digestion? Well, they used to think that the stomach was purely kind of mechanical. It just churned food. And they didn't know that there was chemical digestion. They didn't know that there was the release of enzymes or really acid. So what happened to this guy is he got shot in the stomach. He got shot in the abdomen, and that left a hole. It left a hole that didn't really heal that well. And he was basically used as a kind of scientific experiment. And it's a really interesting story. The doctor who was using it as a scientific experiment, definitely not ethical by modern standards. And so what he'd do was he'd look inside the hole and he would take samples. And from doing that, he managed to, I think it was quite a few years, he studied and he managed to figure out that there was chemical digestion going on. So this is a case of what we call serendipity, which means a lucky occurring, a lucky happening. I suppose not lucky for the man, but lucky for science that he was shot in the stomach, and that when it was stitched up, it healed in a way which allowed the stomach contents to be studied, and it showed that chemicals were released into the stomach and it wasn't just mechanical digestion. And that's the end of the first part.